So thank you so much for inviting me to give this talk. I'm really honored to speak in front of you all. My name's Becca. I am a PhD candidate at the University of Utah, and my project is on kidney disease and ceramides. Kidney disease is prevalent in the United States. It affects about 1 in 7 US adults, and about half of ICU hospitalizations are complicated by acute kidney injury, or AKI. And these are really burdensome conditions in terms of patient morbidity, mortality, and quality of life. And they also really disproportionately affect our US healthcare spending. So for example, our Medicare population is about 15% made up of individuals with chronic kidney disease or kidney failure, but those patients account for about 1/3 of total Medicare spending annually. And importantly, this population of individuals with kidney disease is growing, and there's a huge demand for the discovery of targetable mechanisms driving kidney disease to inform novel therapeutic development. So to orient you to the kidney, we have here a kidney cross-section and blown up a nephron, which is the functional filtering unit of the kidney. So kidney filtrate is generated by the diffusion of fluids, solutes, and toxins from the blood supply in these glomerular bundles into the tubule. And this segment of tubule directly adjacent to the glomerulus, the proximal tubule, is responsible for the majority, reabsorbing the majority of kidney filtrate solutes like salt, glucose, and amino acids back into the blood supply. And that's a really highly energy demanding process. So the cells lining this proximal tubule rely really heavily on the aerobic oxidation of fatty acids to generate ATP. And importantly, it's come to light that metabolic insults to this proximal tubular epithelium, including mitochondrial dysfunction and altered lipid metabolism, are key determinants of kidney disease, regardless of the underlying cause. And they're tightly coupled to clinical endpoints like kidney fibrosis and decline in kidney function, both in terms of the progression of chronic kidney disease and the transition to chronic kidney disease following an acute kidney injury. So for the remainder of this talk, I'll present our preliminary work that the bioactive lipid ceramide is implicated in kidney metabolic maladaptations, driving injury and disease. So I have here the 4-step de novo ceramide synthesis pathway, beginning with the condensation of palmitate and serine to form the sphingosine backbone. Next, the addition of a variable acyl group by one of 6 ceramide synthase enzymes. And lastly, the incorporation of a delta-4 double bond by dihydroceramide desaturase to convert a dihydroceramide to a ceramide. And I want to highlight this step because we have data, and I'll highlight some of my own today, that this double bond distinguishing a dihydroceramide and ceramide confers a big difference in the lipid's pathological fate. So ceramides, as you know, can then be further processed with the addition of head groups to form complex sphingolipids like sphingomyelin and hexosyl ceramides, or they can be degraded by the family of ceramidases or the ceramidase activity of the adiponectin receptors. And in bold text here, I have all of the genes and coding enzymes in this pathway, and you'll see a lot of the same names popping up here on this table. So we've gotten some exciting data from one of our collaborators, Dr. Katalin Sustak at the University of Pennsylvania, that suggests that dysregulation of ceramide metabolism transcriptionally in human kidney tubules correlates with kidney disease endpoints. So these data are generated from a microarray of tubules microdissected from 356 patient nephrectomies. We ran Pearson's correlations to assess the linear relationships of these tubular ceramide-related gene expression with kidney fibrosis or estimated glomerular filtration rate, which is a surrogate measure of kidney function. The color scheme here indicates the magnitude of these correlations and whether they associate with a healthy kidney in green or a diseased kidney in red. And we were surprised by how many and how strongly some of these genes correlated with kidney disease endpoints. Specifically, we were interested to see that this group of genes that positively correlates with worsened kidney outcomes were enriched for genes involved in ceramide synthesis. And alternatively, the group of genes that correlated with favorable kidney outcomes uniquely contained genes involved in ceramide degradation. And so our simplified interpretation of these data is that the accumulation of ceramides could exist along this continuum of worsening kidney disease. And I'll also just point out some interesting divergent associations that we observed for ceramide-related gene expression encoding enzymes that perform similar sphingolipid-related reactions. And we attribute these divergent associations either to the length of the variable acyl chain as seen by these divergent associations of SIRS2 and SIRS5, or potentially the subcellular location of the lipid products related to their enzyme's pH optima or the subcellular location of the enzyme itself. So we wanted to know how these associations of ceramide-related gene expression in kidney disease translated to absolute ceramide levels in diseased kidneys, and we were generously donated some kidney cortex samples from the SUSTAT group to measure ceramides. On the y-axis, you're looking at lipid concentrations, and on the x-axis are individual ceramide species distinguished by their variable acyl chain length or total ceramides. The left model of kidney disease is the folic acid nephropathy model. So that's a toxin-mediated kidney injury. On the right, we have the unilateral ureteral obstructive surgical model of kidney disease. In both cases, we see differences in ceramides. So for folic acid nephropathy, kidney cortex tissue had higher levels of C16 ceramides that are a particularly deleterious species. These UUO kidneys had increases in several individual ceramides and also an increase in total ceramide levels. And that gave us an exciting rationale to manipulate ceramide levels in vivo in a model of kidney disease. So we employed this bilateral ischemia reperfusion acute kidney injury model, it mimics the mitochondrial fragmentation and respiratory impairments, as well as the clinical endpoints observed in humans. And the way we operate this model is we clamp blood supply bilaterally to the kidneys for 22 minutes to induce renal ischemia. And then we remove the clamps to initiate reperfusion. And we utilized animals that harbor a tamoxifen-inducible global ablation of DEGS1, encoding this last enzyme in the ceramide synthesis pathway to inducibly lower whole body ceramides. So for this study, we have our 4 study groups made up of the DEGS1 inducible global knockout animals, or flocks controls that sustained a kidney injury, an ischemia reperfusion injury, or a sham operation. We induced gene deletion prior to the kidney injury and euthanized animals 48 hours after surgery. And first, we confirmed that our whole body knockout is also effective in the kidney and effectively lowers kidney cortex ceramides. But here what we're looking at is plasma urea levels following surgery. High blood urea nitrogen is a clinical marker of kidney disease. And what was really exciting for us was that the knockout animals who received an ischemia reperfusion injury in blue were protected from this spike in plasma urea levels observed in the flocks control animals 24 hours after surgery. So to start looking for a mechanism for this protection with ceramide lowering, we've started to characterize mitochondrial respiratory impairments in cultured human proximal tubular cells caused by an induction of ceramides. So first, I'm treating the HK2 cell line for 2 hours with the cell-permeable C2 ceramide or C2 dihydroceramide in red and blue, or vehicle in black. And after treatment, we run these cells through a mitochondrial stress test on a seahorse analyzer. And we're looking at whole cell oxygen consumption rate at baseline, after inhibition of ATP synthase with oligomycin, after stimulation with the uncoupler FCCP, and after termination of the of oxidative phosphorylation with the complex 1 and 3 inhibitors known in antimycin A. So you can see at baseline and under maximal respiratory conditions, we see decreased oxygen consumption in the C2 ceramide treated cells. And this is just reiterated here on the right, in addition to an impairment in ATP production and spare respiratory capacity inferred from the seahorse trace. Notably, there were no impairments elicited by treatment with the same concentration of C2 dihydroceramide. And again, although those 2 molecules only differ by a double bond, the pathogenic potential of ceramides versus dihydroceramide, here shown as their ability to impair mitochondrial respiration, seem very distinct. In a separate experiment, I induced ceramides instead by treating cells for 24 hours with incubation in palmitate-containing media, palmitate being the primary substrate for that de novo ceramide synthesis pathway, with or without the addition of myriocin, which is a pharmacological inhibitor of the initial enzyme of the pathway SPT to prevent ceramide accumulation, as shown here. Again, we see impairments in oxygen consumption at baseline and under maximal respiratory conditions with the induction of ceramide. And what's exciting is that we prevented mitochondrial impairments associated with palmitate treatment by inhibiting ceramide synthesis. So key takeaways today. First, I want to stress that and reiterate that transcriptional regulation of ceramide metabolism in human kidney tubules correlated with kidney disease endpoints, and specifically the upregulation of ceramide synthesis in human kidney tubules seemed to associate with kidney disease, and ceramide degradation could be renoprotective. Second, the ceramides are elevated in diseased kidneys in preclinical models, and that global genetic ablation of ceramide synthesis prevented kidney injury following renal ischemia and reperfusion. And third, that this effect could be related to an exciting novel mechanism of ceramide driving mitochondrial dysfunction in the kidney tubule. So these have exciting implications for us for understanding the mechanisms of kidney disease and could also inform future therapeutics. So I want to thank my mentors, Drs. Scott Summers and William Holland, as well as the rest of our lab group, and really important collaborators at the University of Utah and elsewhere, as well as members of our University of Utah core facilities. Thank you guys for listening, and I'm excited for questions.